Welcome students. This is for Pharmacognosy and Phytochemistry 2 for the 5th semester B Pharmacy students, and it will be useful for the other students also because it's common extraction method, modern extraction method. The chapter describes in various parts and very interesting chapter. So this is only lecture 1 which I want to explain it about the modern method of extractions and the general methods of extraction. It needs more lecture. Basic of phytochemistry also needed, which I have given some chemical test which can be identified by phytochemicals present in the plants. It's very important for the pharmacy students and 5th semester students, Pharm D students, and one who is undergoing research of identifying phytochemical investigations. So even in this chapter we have to discuss about various spectroscopic methods, chromatographic techniques, including electrophoresis, how it is used for purification, identification, isolation, and characterization of the isolated compounds from the natural sources. So we had this class, but we should have to discuss about this lecture. Before this lecture, in unit 4, from basic phytochemistry, modern method of extraction, applications of latest techniques like spectroscopy, chromatography, and electrophoresis in the isolation of herbal constituents, purification of herbal constituents, identification of herbal constituents of crude drugs, in which the first question in July 2024 recently, they have asked from this question, many questions they have asked. So the students should have an idea about in KUHAS and as well as from the University of Kerala University of Health Science, what all the past questions they have asked and how depth we can study in this chapter plays the major role. So that's why I have just taken all the questions to be focused to the student before starting the lecture of this unit. The question which is asked is explain the Soxhlet extraction, continuous hot percolation example Soxhlet extraction with a neat labeled diagram. So it is very important for the students to write about the condenser, Soxhlet apparatus, about the siphon tube and the side tube, and about the round bottom flask, and even about the mantle or the hot plate which you are using for heating the solvent which you kept in the round bottom flask. So it is very important if a student is known to the practically if you are doing in the lab, it's available in our lab many Soxhlet apparatus in the sizes and the capacity variations are also available, which it will be more helpful if the student go and do it as an experiment for extraction by hot continuous Soxhlet apparatus. And also it's an example for hot continuous percolation method, Soxhlet apparatus. And you see this question again in the July 24, they have asked from this chapter, what do you mean by edge effect in TLC, thin layer chromatography? So you should know about the chromatographic technique, about thin layer chromatography, paper chromatography, column chromatography, HPLC, high performance liquid chromatography, gas chromatography, and then high performance thin layer chromatography. So there are many other chromatographies like gel permeation chromatography. These all have to be studied in detail, gas chromatographies. And also about spectroscopic methods. What is its application for the herbal formulations and also herbal drugs, isolations and characterization of the compound. You should know about various spectral methods, UV, IR, NMR, mass, and even about the many other emission spectroscopy also needed for you to know about certain things. This is just a picture I am just giving you about the edging effect. We will have a full detailed discussion when I am explaining about the thin layer chromatography. Define chromatography, 2-mark question. The same question in July 2024. You just imagine they have asked how much important this chapter. And principle of electrophoresis, 2-mark question. In the same question, July 2024. And a maceration, 2-mark question, July 2024. You should tell about different types of maceration. Define maceration not only sufficient, you should write different types of maceration, which last class we have discussed about that. Even in this lecture also I have included about the maceration. Modified bone trigger test, which we might have studied in androgen glycosides in Senna. When you are studying, it's an important test for modified. Bone trigger test is also there, modified bone trigger test. So chemical test for all phytochemical investigation comes in the chapter. It is very important. Already I have delivered one lecture in online. It is necessary for the students to listen to that and to note it down what all the chemical test which is needed for us to write it clear and to study them and to express in the examination point of view. And you can see explain the role of chromatographic techniques in isolation and estimation of herbal drugs. 10-mark question. So the student should write detail about thin layer chromatography, paper chromatography, column chromatography, HPLC, HPTLC, gas chromatography techniques, and starting from the definition and classification and their principles and what is its application towards herbal drugs, and write some examples for this 10-mark question. So you should know about this chapter is having that much importance about various types of techniques and the principle behind. Give any 5 application of spectral methods, 2-mark question. So simply if you write application of UV to herbal drugs like that, it will not be useful. What drug, in what nanometer, what type of absorbance have been used in detection of UV spectroscopy, and what type of application of IR spectroscopy can be applied for a herbal drug to find out a functional group, and what type of application of NMR which can be used for detection of number of carbon atom, number of nitrogen, number of hydrogen. Like that there are many NMRs, and mass spectroscopy, how it can be used for finding out the mass number of the component which is isolated from the herbal formulation or the herbal drug or a herbal plant or a herb which you have taken and isolated by means of chromatographic technique, and then study for characterization of spectral methods should be explained very clearly. Even though if it is 2-mark, you should write at least any one example is very much needed. It's an important question which they have asked. And number 9, what is supercritical fluid? What do you mean by supercritical fluid? Means immediately students should know that the critical point in your physics which you have studied about solid, liquid, gas having a critical point where a substance can be stand in a critical point, a liquid or a solid, and also gas, it can be prevent in a temperature and pressure when you control. So you know that carbon dioxide, liquid carbon dioxide using as a critical fluid for extraction is a new technique which is an advanced technique and easy for removal of the solvent system. These all advantages are there. There is experiments, there is examples and explanations are given separately for this supercritical fluid usage of extraction method using carbon dioxide, liquid carbon dioxide. So that is December 2023. They have asked, which application of gel write the application of gel electrophoresis. Only 2 mark they have asked. Gel electrophoresis is a very important technique for DNA fingerprinting, even for identifying the DNA samples and identify the person of who is responsible, who is having the similar DNA of a particular sample which is taken. A tissue can also be identified. So it's a very good, interesting forensic technology which belongs to this electrophoresis, gel electrophoresis, and how it is applied for an herbal formulation, herbal techniques to make them, to find out which herb contain the particular constituents can be identified by means of this electrophoresis. 2 mark question. It's an interesting subject which you want to explain about detail and describe in detail about column chromatography. We will be having a discussion about 5 mark question they have asked in September 2023. Describe microwave assisted extraction method. Microwave. Using the coil of microwave, you know microwaves in the home, how they are using for the cooking substance. And here microwave techniques can be used for even extraction of volatile substances, volatile oils. We are using microwave technique which can be assisted. So they have asked 5 mark question. The student should have the capacity to write. Even I have explained detailedly about in volatile oil chapter, which I have shared, and you can just, I will share today and also to the group, and the students are necessary for you to go through volatile oil, what all the techniques of extraction, in which this is one of the important technique of microwave assisted extraction method. Write the application of IR spectroscopy in herbal formulation or herbal isolation techniques. So you can see in a specified IR spectroscopy is used for finding out the functional group, whether it is a ketone, aldehyde, nitrogen, amine group, alcoholic group, or even whether it is having what type of ring structure can also be identified by means of this. Functional group can be easily identified by means of infrared spectroscopy. So that is also plays a major role to identify or characterize the isolated compounds. So you can see that is 14th question in 2023 May. They have asked principle and application of high performance thin layer chromatography. It's an important technique. Most pharmaceutical industry, they are now depending on this HPLC, high performance thin layer chromatography, for various assays, content variations, content identification. All these things can be used, even formulated substances and also the fresh crude drugs and generic compounds can also be identified by means of HPTLC. HPTLC is one of the important technique, high performance thin layer chromatography, for various separation of thin layer and also more sophisticated, more interesting material which can be determined. These are all the instruments which is needed for a HPTLC. We will have a discussion about HPTLC. Principle and application of electrophoresis, 2 mark question, May 2023. Application of mass spectroscopy. By means of ionization of the molecules, you will be having the detector which can detect by means of electromagnetic force, or you can see electric generation by means of the ions, the daughter ions, and what is the mass number of the particular compound can be identified. So mass spectroscopy, IR spectroscopy, UV spectroscopy, NMR spectroscopy, these all together a person can identify the structural molecules or elucidate the structure. You can say elucidation of structural compound of a natural compound can be easily identified. So we have to study about the general mass spectroscopy application. Modified bone tracker test, again they have asked in 2023, May 2023. Explain chromatographic techniques used in plant drug analysis. 5-mark question. So if the student knows all the techniques which is belonging, different types of techniques of chromatographic techniques, there will not be any problem. A student can explain how much extent he can explain according to 5-mark or 10-mark or 2-mark. According to that, you can go for explanation. Mention the spray agents used to detect alkaloids. So if you know, especially the chemical test for alkaloids like drag and drop reagent, Hager's reagent, Wager's reagent, these all reagents are coloring agents. So usually drag and drop reagents. So spraying reagents which can be used even for detecting the spot in the HPL, in the TLC or HPTLC, they are asking what, mention the spray and what is the chemical name of the spray. You cannot write only drag and drop. Drag and drop is a scientist name. So you should write what is the chemical name. What is the mercuric potassium chloride, or what will be the chemical name should be mentioned very clearly. That's what they mean, that is for 2-mark question. Outline of the role of IR spectroscopy in evaluation of plant constituents. What is the evaluation? It can find out what type of functional group is present, and detailedly you can write an example also. Discuss the detailed modern methods of extraction of phytoconstituents. 10-mark question. So the student should write all the basic techniques of extraction and the modern techniques like for volatile oil techniques, the names and the explanation. And also you should write about the liquid carbon dioxide, critical point, fluid, gas, carbon dioxide, liquid carbon dioxide using extraction can also be explained. Vitali-Morse reaction is a chemical test for alkaloids, specific test, 2-mark question, we can write that specific test. So all the chemical test for phytoconstituents is very, very important, name reaction. Killer-Kilani test for cardiac glycosides, which you might have studied now. This is also a phytochemical investigation chemical test. So I made a lecture all the phytochemical tests which you have studied, basic starting from carbon dioxide, from carbohydrate, lipids, though it is very much even tannins, flavonoids. So it's very much important for a pharmacy student, and especially the 5th semester student, you should be thorough with that. That is why basic phytochemical investigation, the student should know means all the chemical test, all the phytochemicals whichever present, basic phytochemicals of secondary metabolites, the names should be very clearly known for the student. So now the today's class we will have, still more questions are there. That is principle involved in paper chromatography in November 2021. Summarize the use of electrophoresis in phytochemical analysis, 5-mark question, January 2021. And you can say describe the principle involved in gas chromatography and the application from the pharmacognosy. So how a gas chromatography works with the long column, which is twisted column which is shown. I will explain about this chapter very clearly. And also in a short form how it is application and its principle is needed for the student should know. And 27th question, elaborate on various chromatographic techniques for isolation and purification of phytoconstituents. 10-mark question in January 2020. You just imagine, it is a 10-mark question. So if a student knows only TLC or only paper chromatography will not be sufficient. 10-mark question, you should write all the techniques regarding what all the application of the techniques. If you explain, then only the evaluator can give you 10 out of 10 marks. And principle and application of thin layer chromatography, specifically they have asked 2-mark question. So you should know what is the importance of 10-mark question and not writing only the simply a principle, and if it will not give you 2-mark for you. Gambier fluorescent test is a very important test which you can see in pale Katachu. All the you can see that is for this also a name reaction according to the phytochemical substances. which is I have explained all the things in the phytochemical test, chemical test. And you can see combined amblyophoron test, 2-mark question in 2020 they have asked January. So, and Mirexite test, which is used for identifying caffeine, pseudoalkaloids, which will not test for other test will not be answer for caffeine, xanthine alkaloids. So that's why we have to go for Mirexite test. So these all tests will come in the phytochemical test which I have given a class. So students are requested to, are informed to listen to that class, note it down all the chemical tests and do the needful for your own, making yourself comfortable for writing the exam. I feel it's very important. Now we will go to the small class which is for the lecture 1 regarding modern method of extraction. I cannot go for the complete extraction today, but still I can come up to the Soxhlet apparatus because it is needed for you to explain today about the Soxhlet apparatus which already. So when you talk about extraction, very simple, the definition should be clearly explained. This I have taken from a book which is published by a very important teacher. He has written very clearly. In the extraction process, maybe animal source or a plant source, the drug is treated with particular solvent. What do you mean by the particular solvent? So solubility of the component plays a major role. Particular solvent which can solubilize the compound which you are interested. The solvent dissolves the medicinal active components or constituents in itself, but animal and plant tissues and other components are not dissolved in the solvent. We need only the active principle. So we have to select such a solvent system which can solubilize the compound which you are interested. In other words, you can say the definition in other words, extraction is the process in which the separation of the soluble constituents occurs from insoluble substances, either solid or liquid, by processing with a specific solvent. Extraction of plant materials, some of the points. All plant materials are first has to be properly authenticated. So time and money is not wasted if the person is not doing authentication in the beginning itself. Examining material or a doubtful origin after doing your experiment, it will be avoided if you do the authentication by proper botanist. The choice of extraction procedure depend on the nature of the plant material components to be isolated. Dried material is normally powdered before extraction. Fresh plants, leaves, flowers can be homogenized or macerated with a solvent. Example, alcohol. Alcohol is also used or useful for stabilizing fresh leaves also. Solvents used for plant extractions. Alcohol is a general solvent for many plant constituents, except there are many exceptions are there. Water immiscible solvents are also commonly used. Example, light petroleum ether, volatile oil and fixed oils and steroids can be used by light petroleum. Ether and chloroform can be used for alkaloids and quinones. So already in the class I have told very clearly, you should know at least the list of solvent systems starting from hexane. Increasing polarity, when you see hexane, petroleum ether, benzene, chloroform, carbon tetrachloride, and then comes acetone, methyl acetate, ethanol, methanol, water, dilutes HCl, dilute sulfuric acid, dilute nitric acid. So like that you can increase the polarity for solvent system to make your extractions. Extraction of alkaloids, phenol. When you come to phenol extraction, before extracting of alkaloids and phenols, when you want to extract alkaloids, you know that alkaloids are nitrogen containing compound, basic in nature. Basification of the plant is necessary before extraction. if a water immiscible solvent is used. If it is you are going for extracting of aromatic acids or phenols are going to be extracted, acidification may be required. Method of extraction process for phytoconstituents, various methods of extractions are there, starting from infusion, decoction, digestion, maceration, countercurrent extraction, supercritical extraction, hot continuous extraction, Soxhlet apparatus or Soxhlation, percolation, ultrasound extraction, steam distillation, microwave extended extraction. And now some of the definition for the extraction the student should know. In the classroom also I told. Like when you go for filtrate or filtration, in your pharmaceutical engineering, you might have studied about size reduction, filtration, extraction, all these things you might have studied. So there are some terms you should know about that. For filtration, you should know what is filter medium, what is filtrate, what do you mean by residue. Like that you should know some of the words which you want to write about filtration process. So same thing when you want to go for extraction, you should know what is menstruum. Menstruum means solvent or mixture of solvent used for extraction. Very simple. The solvent, the name when you are coming for extraction of crude drugs in the plants, in a herbal formulation or a herbal drugs, then you can say menstruum. Micella. Micella means the solution with extracted substance. So after extraction, when you get the filtrate or when you get the compound in the solvent, that is called micella. So you should know some of the terms. And rinsing, we used to use rinsing, word which you are using in the laboratory also. So here in extraction, dilution of extractive substances out of disintegrated cells, out of the coarse cells, out of the coarse powder cells of the plants or the animals. Laxative or leaching. What do you mean by laxative or leaching? Laxative. Short notes they may ask, what do you mean by laxative or leaching? Water is used as a solvent for extraction. When you use solvent, selection by means of water, a common high polar solvent, water when you are using, it is called laxative or leaching. Mark, press the mark, you might have studied in your pharmaceutics. Mark, residue left after extracting after extracting the desired constituents, desired the chemical constituent, desired the medicinal components from the crude, that is called the residue, the remaining residue, that is called mark. Infusion, normally prepared in our home, normally available, very easy, just without shaking, in a solvent, water keeping ginger infusions which we prepare in our home, which we are using. So in the infusion process, the crude drug is left with solvent to soak them properly at room temperature for a particular period of time with or without intermittent shaking. After the specific time period, the filtration will allow, which will separate the plant or plant's debris, unwanted substance, with the micella. Micella means what? The solvent which contains the active principle, that is infusion, which is prepared. Various types of plant material has settled down, which has separated by decantation and can replace with other solvent. Decoction, coffee which you are preparing in the home. Simple example, take drug in a vessel, add 1000 ml of water and cover it, boil for this for around 15 minutes. Cool it to room temperature, strain the liquid and press the mark. Digestion, you have to digest, you have to heat, you have to make them to be digest, complete the cells to extract. So digestion is modified form of maceration in which the temperature of the menstruum is kept heated at particular temperature at under pressure. This rise of the temperature and pressure help the menstruum to penetrate the drug and extract it completely. The apparatus used for the digestion is the digester, which is generally made up of metal. The drug is placed inside of it with menstruum under specified temperature and pressure, and then screw the cover with the help of nuts. Maceration. Many times they have asked the questions. In the process of maceration, the crude drug is immersed into the bulk of menstruum, solvent or solvent. Menstruum is the other word for solvent. For at least 3 days, generally 3 to 7 days. During the period, the menstruum is agitated frequently. The menstruum and container should be kept in a stoppered container. The mixture is then filtered or strained through net or sieves. The maceration may be of various types like simple maceration, modified maceration, multiple maceration, kinetic maceration, remaceration. So when you come to the comparison of simple maceration and modified maceration, very simple, the student should remember about organized drug and unorganized drug. Organized drug means we can go for simple maceration. Unorganized drug means modified maceration. You should know that definition which you studied in the 4th semester. So, use of organized drug, bark, root, stem, like that you are using. Use of unorganized drug like exudates, like gums, resins. And the drugs of the menstruum shaken occasionally during 3 to 7 days. Here, as it is an unorganized drug, cakes may be formed in the down bottom. So, drugs and the 80% of menstruum shaken occasionally during 3 to 7 days. Strain the liquid and press the mark. But you can see decant the liquid, mark is not pressed here. And mix the liquid obtained and clarify. Here in the modified maceration, filter one liquid and pass remaining menstruum. And the point in the simple maceration is filter is not adjusted to the volume. But here in modified maceration, where you are using the unorganized drug filter which you received, that is mesalline, is adjusted to the volume. And you can see number 6, the point which is already we are told about this organized drug and unorganized drug. It is a repetition. Anyhow, it is also an important point for the 6th, the common. Here, the mark is pressed because contain considerable amount of macerate in as it is an organized drug. Here, in the modified maceration technique, mark is gummy and compact and do not contain any macerate. So pressing the mark is not necessary as it is an unorganized drug. Multiple maceration. The maceration process is used the concentration preparation where entire maceration menstruum is segregated into 2 parts. For double maceration or 3 parts in the triple maceration, each menstruum are used individually for the maceration process. In this process, the drug menstruum ratio is low and the extraction is performed with less amount of menstruum. Kinetic maceration. Like the simple maceration, kinetic, you are giving some energy. Kinetic maceration is also proceeded into the room temperature. But the single difference is that the material is under constant motion. The intensity and the type of movement plays important role in the maceration. Remaceration. In the remaceration process, part of the solvent is added to the drug. After filtration, process the residue is treated with the remaining solvent. Use of maceration. British Pharmacopoeia permits the preparation of tincture by maceration method of the following drugs: squill tincture, catechu tinctures, senna liquid extracts, compound benzoin tincture, and opium tincture. Mother tinctures of homeopathic pharmacopoeia are prepared by maceration methods. Maceration is widely used method of extraction. Maceration can be performed easily with pharmacist lab and a small amount of sample. There are number of drugs which can be extracted by only maceration method due to their high swelling property or high mucilage content. Percolation. Immediately you should know about this picture, menstruum, and how the filter paper is packed, and the drug, and here the glass wool, and with a tape. Percolation means it is a passing through. The word percolation refers to first pass through, the solvent or a menstruum pass through. Percolation can be defined as short successive maceration. Short successive maceration or extraction by method of displacement. The solvent is displaced by a new one. Percolation can be performed into conical percolator, cylinder percolator, and steam jacket percolator. The same percolator can be having a steam jacket also. So the various stages involved in the percolation are: size reduction first, imbibition is second one, packing. Size reduction, you have to reduce the size. In the classroom we have discussed about the size reduction which you studied in the engineering. Size reduction, cutting, compression, attrition, impact, attrition. These are the four techniques which you have used. Many instruments you might have studied, especially ball mill, have undergoes all the techniques except cutting. It undergoes impact, attrition, and compression. It's a very good techniques of the using size reduction, which can increase the surface area, improve the extraction process, improve the efficiency of the extraction process. Imbibition with the solvent, wetting, making the component, making the material to be ready for extraction. Packing. After imbibition, the lump, made into a lump, the crude drug must be broken. The lower end of the percolator should be plugged with a cotton and then placed the drug powder layer as shown in the picture, layer by layer. The packing should be perfect. It should not be neither too tight nor too loose, which may affect the flow of the menstruum. You know that menstruum or the solvent system have to have a free movement to extract the main components. 2/3 of the percolator should be covered with the drug, on which place a piece of filter paper, and washed clean sand should be placed on the top of the filter paper. This prevents any type of disturbance of the crude drug by flow of menstruum. So, the next step is maceration in the percolation. What is happening there? Sufficient amount of menstruum should be added from the top after packing of the percolator. During the addition of menstruum, the lower tap of the percolator should be open for some time, because the air which is trapped can be removed and the percolator can be displaced by the menstruum in the air. When menstruum starts to come from the lower tap, close the tap and allow to stand the percolator for 24 hours as the primary maceration takes place. The menstruum level should be above the drug bed as shown in the picture. Last one, percolation. Open the lower tap after 24 hours, the maceration, and collect the menstruum from the lower end until the 3/4 of the portion of the final product is obtained. Meanwhile, the sufficient menstruum should be added over the powder so the packing drugs does not become dry. This can be repeated. Completion of the percolation process should be checked by the various method. Whether it is completely extracted or not, how will you identify? It can be identified by checking the specific gravity of the end few ml of the percolate or the amount or the menstruum after extraction obtained, or micella, or which you got percolate is compared with the specific gravity of the fresh menstruum, fresh solvent, and evaporate the last few ml of the percolate to dryness and see that any residue comes after evaporation or not. Perform the specific chemical test for the last few ml for the percolate, like tannin test, glycoside test, alkaloid test, etc. That means all phytochemical investigation tests. So the student should know chemical test in the first itself from the chapter before extraction. If it is positive, go for the again repeated percolation. If it is negative, you can stop your percolation process. Now press the mark. Don't lose any solvents or any menstruum remaining in the drug molecule or the coarse powder molecule which you used in the percolator. Press the mark. The mark should be pressed and obtained. The liquid is added into the collecting menstruum. More menstruum should be added to obtain the desired volume. Allow the liquid to stand and separate the suspended particles by filtering or filtration or decantation. Example: strong tincture of ginger, tincture of belladonna, etc. are the example which is prepared by this conical percolator. Modified percolation. In modified percolation process, we reduce the drug percolation or percolate ratio 1:3, which is generally 1:4 in common percolation process. By this, we can save menstruum time and heating. Percolation is type of displacement method. It has been observed that a stationary menstruum dissolve the more active constituents because stationary menstruum may remain in contact with more time to drug compared to the moving menstruum. Soxhlet apparatus, which is very important, the question have been repeatedly asked. Draw the diagram of the continuous hot percolation process. This is the Soxhlet apparatus. This is the picture. You see, I have not given any explanation or written in this slide or notes. Students should understand and listen to the lecture, and they should be able to write an essay or even a short notes. 5 mark, 10 mark or even 2 mark, it is possible for you to write. You see the picture very clearly, number 1 and number 3 and number 5. So number 1 is round bottom flask and number 8 you can see it is a heating mandle. So the student can write starting from the number 8 from the bottom. A heating mandle is needed where the round bottom should fix over there for heating. Round bottom flask, the number 1, according to the capacity of the requirement, 1 liter round bottom or 500 liter round bottom or 2 liter round bottoms flask can be used according to the capacity of the Soxhlet apparatus. The number 3 is another separate attachment which is called Soxhlet apparatus. In the original picture also you can see very clearly. And number 5 is condenser, water condenser. Every student should know water condenser. You can see the arrow in the picture. One is inlet, another is outlet. The top is inlet and the down it is coming outlet. So outlet. So the liquid or the water which circulate in the water condenser cool that area, condensing area. So you see here there is a number 6. What is it? It is siphon, S-I-P-H-O-N, siphon tube. And 7 is a side arm tube. You should observe very clearly the round bottom flask is having the solvent, and when it is heated by the heating mantle, the the vapors, the solvent may be any selected solvent, maybe ethanol, maybe methanol, maybe even you can go for low polar solvents like chloroform, or even you can go for petroleum ether, even you can for methyl acetate, whichever is you are needed to use and comfortable for your active principle to be solubilized by the hot continuous percolation method, you can select the solvent. So when you heat in the number 8 heating mantle, which is kept in the round bottom flask, the solvent, which will get heated and evaporate, and see that it will travel through the side tube number 7, because you see in the 3 there is a line, it is closed, the vapors cannot pass through. If the side tube, that 7 is the side tube, side arm, by means of glass tube in the Soxhlet apparatus. So it travels and comes to number 4 area and goes to the number 5. So what happened? Once it enters into the number 5, there it is cooling water condenser, the solvent will get condensed. And when it is condensed, it becomes liquid, hot liquid, falls on the drug which you kept in the area number 3. Number 3 is the place where we are packing with the drug, crude drug, which you are interested to extract. So you should write very clearly, draw the diagram and very clearly write the solvent is present in the round bottom flask and the crude drug is packed, even which is it can be a packed with a bag we can keep, or directly also we can pack. Number 3 is the place where the drug is kept, which you are interested to extract. And by means of, in the number 4, the ground joint, in the borosil ground joint, you are fixing one water condenser, that is number 5 as shown in the picture. So you can see the hot solvent, after condensed it will come on dripping over the position number 3. And once it drips, you can see below 3 there is a line, so there is no chance of the solvent directly coming back to the round bottom flask. So it started filling, it started soaking, it started go on increasing the level in number 3 area. So once it comes to the level equal to the siphon tube shown in the number 6. What is number 6? Just see the picture. Number 6 is a siphon tube. So siphon tube, you can see the picture very clearly. The siphon tube is connected in number 3 area of the Soxhlet apparatus. I am repeatedly telling, number 6 siphon tube is having a connection in number 3 area as a bend tube like this and connected. You can see that very clearly. Only the siphon tube is having connection in number 3 where the drug is kept. So in that, so when the solvent rises in number 3, the solvent in number 6 also it will rises. Once it goes to the same level, reaches to the maximum level of number 6 in siphon tube, it siphons. So siphon means the number is, according to the Pascal's law, you should know that siphon, it drains the solvent back to the round bottom flask. It siphons once it reaches to the maximum level. Once it is siphoned, it removes all the solvent. You can say it removes or it sucks or it brings down to the round bottom flask in the number 1 back from the 3. Completely it will drain. And it coming back to the round bottom flask. And then again there is heating is going on by means of the mantle, the round bottom flask. The solvent alone will be evaporated, continuous evaporated, goes through the 7, reaches to the 5, condensing area of the water condenser, come back to number 3, and slowly again going the level to the top in 3 and coming to the maximum level of number 6 and siphons back to round bottom flask. So this is a continuous process. So usually they will do 72 hours and they will watch how many times the siphons, how many times the cycle taking place. These all can be parameters, can be noted down when you are doing the research. So the temperature may also be controlled and you will get a very good extraction and go for various chemical tests, identification, and we can perform pilot, small amount type of smaller Soxhlet also available, larger capacity, even 1 liter size of capacity of Soxhlet apparatus are all available. Increasing the round bottom flask also up to 2 liters, it is possible for you to increase the solvent system. Bigger water condensers there can be prepared. So it is very important for the students to write for 5 mark or 10 mark. Write very clearly, Soxhlet apparatus comes under a continuous hot percolation method and it has 3 parts mainly, and we can add in the picture 4 parts, but apparatus have only 3 parts, specifically round bottom flask with a ground joint with Soxhlet apparatus and with a ground joint attachment with an water condenser. The 4th one which I told is heating mantle, and even 5th one you have rubber tubes which has to be connected for inlet for the water and outlet for the water for the condenser which is shown in the picture of number 5. So clearly the students have to write yourself, make your notes and bring to me to my office and submit. That is very important. So it is very important topic for a Soxhlet apparatus. A student should know, and you should prefer to do the practical in your lab, especially in our institution. Try to come and do it. You can ask anytime. I am ready to help you to fix this Soxhlet apparatus in our lab. Many types of Soxhlet apparatus are available in our lab. Thank you. Thank you for being with a small class which I have explained. Listen to the questions, listen to the class, prepare your notes and be ready for the examination. Thank you for being. We will see in the next classes.